Okay. Okay. Student, my pen is not working well, so I am working with my hand. So the topic we have to discuss today is about virus and prokaryotes. That's not working well. Okay. Can I just type here with my hand? Okay. So the topic today is about viruses, moneras, that's also called as prokaryotes. The next topic is about protists. And after that, we will discuss about the fungi. And at the last, we will discuss about the plants. The specific topic is about photosynthesis. And the structure of plant. So first of all, we will discuss about the viruses. Viruses are acellular organisms. Why they are acellular and why they are non-living, they are also called as non-living organisms. And we do not count them in the living organisms. There is a reason behind this. The reason is they cannot produce their own products. And they use the biosynthetic machinery of other organisms. The reason behind why viruses are acellular particles, because they cannot produce their own products and they depend upon the biosynthetic machinery of the other organisms. such as protein or DNA. These are the basic components that a virus requires to reproduce. So it cannot be cultured in any medium, whatever you will provide. So viruses are the only members that cannot be cultured in any medium. Otherwise, all organisms can be cultured in any medium. Just as we culture bacteria and we culture different cell with the help of different mediums. About virus study is called study of virus is called virology. And if we discuss about their structure, now, I am not listening well.
Can you repeat?
I need a repeat for the class, please. Stefan need the repeat. Yeah. The virus. The virus are cellular organisms.
Okay, okay. Viruses are acellular, and these are not included in the living things. Why they are considered as non-living things? Because they cannot produce their own product. They cannot reproduce them itself, and they use the biosynthetic machinery of other organisms. If we culture them, they cannot take the nutrients and do not produce themselves itself. The study of viruses is virology, and if we discuss about their structure, it's only composed of a simple protein and the nucleic acid. And the nucleic acid may be DNA or RNA. About discovery, Edward Jenner was the first who discovered first antibiotics. First vaccine about viruses. Okay, Edward Jenner was the first scientist who created a vaccine about viruses. So, more about them. They are 1,000 times smaller than the largest bacteria. Sorry, the smallest. Their size range from 250 nanometer to 20 nanometer. This is the size in range in which the bacteria in which the viruses can be formed. And the Stanley was the first who crystallized viruses, and the virus was TMV, tobacco mosaic virus. Stanley was the first scientist who crystallized the tobacco mosaic virus, and its simple structure was DNA and proteins. If we discuss about the structure, they are composed of DNA and proteins, and the proteins are called as capsomers. These are the subunits of capsids. Okay, I can create a diagram here. Just a minute. Okay. Viruses.
Alia, sorry, excuse me. The
Okay.
student Epson, I have that one question. No, I
Okay.
don't know, you repeat for the virus topic.
About acellular.
Epson, Epson, what is your question exactly? Etepon. You need to repeat for the class, the virus topic?
Okay.
Okay, Aila. No worry.
Okay, just a minute. Okay, just a minute. I will explain all the things. The viruses are composed of proteins and DNA, and they are considered as non-living organisms. The reasons I explained here is the simple. They cannot reproduce itself. For reproduction or to make a copies of themselves, they need a biosynthetic machinery of the other organisms. That will be the reason, because they are considered as non-living organism or acellular. A means not and cellular, cell particles. So they are not a cellular particles. Okay. Hello.
Hi, yeah. It's okay, Aila. Thank you.
Okay. Okay. So if we make a diagram about the viruses, that's a covering of protein, and here will be the double stranded DNA of the virus that called as nucleic acid, and the protein here is made up of capsomers. Uh, okay. Uh, Viruses have many types. Viruses have many types, and they may be with different nucleic acid, and it can be RNA or DNA. We know that smallpox contain DNA, while the herpes virus contain also contain DNA. While the influenza virus contain RNA. These are the some examples of virus that contain different genotype type or genome. Virus, uh, if we discuss about the disease, virus that eat bacteria called bacteriophage. The virus that eat bacteria is called as bacteriophage, and that will be T4, and it make two types of cycle with bacteria. Either it can harm the bacteria by making a prophage or by lysis. The one is with lytic cycle. The other is lysogenic cycle. In lytic cycle, virus attach its DNA to the bacterial DNA and produce many copies and destroy the cell. While with the lysogenic, cycle, virus attaches its DNA with bacterial DNA and make a prophage for a long time. This is because he needs a multiple copies of its cell. After some time, when the stimulus arrive, the DNA detach itself and again proceed with lysis or lytic cycle. The stimulus can be of can be from the environment, or it may be some sort of radiations from the environment, such as that can be X-rays or UV rays. When the stimulus of radiations or from the environment comes, the DNA of the virus detaches itself from the bacterial DNA, and again it starts the process of lysis. It split the DNA and make multiple copies, and again it destroy the bacterial cell. These are the two cycles that viruses make with the bacteria. Now we will move with disease. The last topic is retroviruses. These are the viruses that contain RNA, or they called as called as oncovirus. The main purpose of these cells to infect the main purpose infect the immunity that will be provided by T helper lymphocytes. These are the cells that provide immunity to our body, and when the viruses enter into the body, it uh, destroys the cells, T helper lymphocytes. The site at which it attack is called as CD4 site. This is the specific this is the specific site or position at which they attack. So we can say that they are host specific. So they are host specific. The virus that infect T helper lymphocytes is HIV. This is called as immunodeficiency virus. This is the virus that attack the T helper lymphocytes, and it attacks on the site that is CD4 sites at the T helper lymphocytes, and again it make a lysogenic cycle with the cell. So
Teacher,
okay, yes,
can you repeat that, please?
okay. Retroviruses are the types of viruses and they contain RNA in it. These are also called as oncoviruses and they mainly infect the immunity of cell. That cell we call as T helper lymphocytes. These cells provide immunity to our body as we called B cells and T cells that infects that kills all the foreign particles that enter in our body. And there is a specific site on these cells at which this attack, and this virus is called as HIV. This is called as human immuno. We can say that these are the host specific because they only attack those cells that have this specific site. We call this CD4 site, and in the monkey, they lack this site. So in monkey, these viruses do not affect them.
Lo escucho entrecortado.
Teacher,
Sorry, excuse me.
teacher is not here very well.
Se fue.
Okay. Now we
No.
hello. Hello.
Hi, Elias.
Hi, Elias. Yeah, we can hear you.
Yes.
Sometimes there's a bit of a lag in the connection. So, when there's a bit of a lag in the connection, you break up a little bit, but let's do the best we can, and let's keep going. I think it's helpful that you're writing what you're saying. So if we can't hear it, we can see some of the points you're covering. So If it gets very choppy, we may ask you to repeat the last thing. Thank you, Elias. Okay, let's give Professor Elias a chance to reconnect. I think he's going to try again, see if we can get a more stable connection. Let's give it a shot. Hi,
Okay,
Elias, you're
Prof.
back.
Hi,
Yes.
Elias, is ready, yeah, the
Okay.
co-host.
Sorry, my internet connection was not good. I'm just trying to connect with my phone. Uh, Now we will move forward with the uh, kingdom Monera. And kingdom moneras are also called as prokaryotes. These are all bacteria and also archaeobacteria. Kingdom Monera only contain bacterial cells, and there are some specific characteristics of bacteria. These are, they contain cell wall of peptidoglycan, while the archaeobacteria do not contain peptidoglycan. All bacteria are heterotropic, and they take their food from the different organism by saprotropic means or by the absorptions. Now we will move toward the different structures of bacteria. Bacteria have different shapes. They may be of circular, or they may be comma shaped, and they may be of bacillus. The oval shaped bacteria are called cocci, and the bacteria with the rectangular shape are called as bacillus. And the comma shaped viruses are called as vibro viruses. Hello. Hello.
Hi. Hi, Ella.
Can you please share the screen? Because it's a topic of that can be only teaches with the diagrams.
May go off. Okay,
Yeah,
Elias, you ready?
Yeah, set him as co-host since he logged off.
You ready, Elias?
Okay, okay.
Okay. Thank you.
So the oval-shaped bacteria are cocci. The rod-shaped bacteria are bacillus. And the comma-shaped bacteria are called as vibro bacteria. And the cocci, if a bacteria bacteria basically bacteria are divided on the basis of their plane of division. And these are maybe of single plane, maybe of two plane, and maybe of three planes. The single plane only division will be on x-axis, on the two axis this will be on x and y axis, and for three planes it will be on x, y and z axis. If a bacteria is let's consider for the cocci, this is the shape of cocci, and if it's only dividing in a simple plane of x axis, this is the x axis. If it's reproducing in the x axis and make a copy in the at the side of x axis, this will be called as cocci, and it is called as diplococcus. Di mean two and coccus mean two bacteria. If it will divide on the y sides, this will be the plane of two axes, and it will divide by this. This is the single bacteria we called coccus, and if it will divide on the two axes X and Y. So this will be the tetrad. This full form is called as tetrad. And if this will divide on the third plane, consider this as a 3D structure. Okay, this is the Z. If it is dividing on the three planes, then this will form as Sarcina. This structure is called as Sarcina. If the division is only in a single plane, it is called as Diplococcus. And if the division is on the two plane, it is called as tetrad. And if the division is on the three plane, it is called as Sarcina. That is a 8 cells made up of 8 cells, 4 on the front side and 4 on the back side. If this only divide in a single plane and make a long chain, just like as this is a single x-axis and division is only on the x-axis and form a long chain, this is called as strepto focus. This is in the form of strip, just like as a thread, and only in the form of x-axis. For bacillus, bacillus is the shape of rod-like, and this cannot be divided on the two axes. Just we call it the x-axis, and this is the y-axis. This is the bacillus, and it only divide on the single plane, and its all divisions can be classified on the single plane. If it is dividing on the single plane, this will form diplobacillus. And if it make a long chain, then it is called as streptobacillus. And this is something like this. This is a single bacillus, and this is the chain of bacillus. If they are dividing in a single plane and making a long chain, this is called as streptobacillus. And the last shape is of comma shaped, and they may be of curve shape. These are maybe of spiro, cheat, or spirillum. The bacteria that are of comma shaped can be of spirochete and maybe of spirillum. The spirochetes are of soft and flexible, while the spirillums are dense and hard form of bacteria. bacterial cell structure. It contains cell wall, and the basic difference between the gram-positive and gram-negative bacteria is doing by the staining. The gram-positive bacteria gives purple color, while the gram-negative bacteria gives pink color. The other difference of the composition of cell wall, and that is the gram-positive bacteria contain they contain peptidoglycan plus lipotachic acids, while the gram-negative bacteria do not contain lipotachic acids. Other difference may be on the basis of their cell thickness or presence of lipids. In gram-positive bacteria, the lipids are of smaller composition, maybe from 4 to 8 nanometer percent, and in gram negative they are from 20 to 80%. In gram-positive bacteria, the lipids compose almost 4 to 8% of their cell membrane or of their cell wall, while in gram-negative bacteria, they only form 20 to 80% of their cell wall. The other structures are maybe of nucleoid. Bacteria do not have proper nucleus in their cell. And their DNA is present in the cytoplasm openly without any membrane. Also, their cell wall do not have cholesterol. The cholesterol is present in the membranes of all eukaryotic cells, while in the bacteria, they lack cholesterol and is present between the membranes of the cell wall. The other structures are mesosomes. These are the membranes that help the bacteria to divide when they make replication. During replication, the mesosomes are the membranes that invaginates inward and separate the two cells. So these are the helper of bacteria in making many colonies. Others are plasmid. This also called as immune responsor of bacteria. When we take any antibodies or medicine, these are the immune to defense, and they make resistance for the bacteria. That's why we call them as the immune functions of the bacteria. Plasmids are immune responsive for the bacterial cells. Also, bacteria form a cyst around their membrane. They make a cyst of different material in different conditions, and that may be due to the environment. They may be of endospores or exosporous. The bacteria that produce the spores inside of their body are called as endospore bacteria, while the bacteria that produce spores outside of their body are called as exosporous. Mode of reproduction in the bacteria is only binary fission. Binary fission is an binary fission is a mode of reproduction in the bacteria in which the two copies of cell formed, and it is a mode of asexual reproduction. Other method can be find and evolution may be due to conjugation, and the conjugation is take place by the pili. Pili are the structures that are present on the cell wall of the bacteria, and they are of hollow appendages. These are hollow appendages on the surface of the bacteria and only used for the purpose of conjugation. And conjugation is a process where the two bacterial cells meet and they transfer their DNA material with one another. The other structure is flagella, and this may be some sort of variety for the bacteria. Consider, okay, sorry. Consider this is a bacteria and this is flagella. If the flagella is present on the one side of the body, this we call this polar side. This is the one side of the bacteria. If a single flagella is present on the one side of the body, this is called as lymphotrichus. Called as lymphotrichus. While if no flagellum is present on the body, then we call bacteria as atrichus.
Elias, please, 5 minutes more, okay?
Okay.
Thank you.
Okay. A means not and trichus means flagellus. The bacteria that do not contain the flagella are called as atrichus, while if there is only a single flagella present on the body, these are called as lymphotrichus. If a tuft of flagella are present on the one side of body, they are called as Tritious flagella. If the flagella are present on the all side of the body, these are called as Pteris trichus. So now we will move toward the protist, and these are the animals that contain unicellular and simple multicellular animals. All the animals that are not fit into the fungi or plantae or animals are put into the protista kingdom. So these may be of animal like, may be of fungi like, or may be of algae like protist. Their resemblance may be found in the cell membrane and for fungi, there may be a resemblance with the cell wall and other structures for the algae that also considered as a plant. So this protist also may be of like algae-like protist. While with the plant, well, the fungi they contain cell wall that may be of animal like because they contain cellulose. And they contain centrioles. Oh, sorry, they do not contain centrioles. So they may be like plants. Plants do not contain centrioles and the animals do not contain chitin in their cell wall. So the fungi have some characteristics that resemble to the animals and some characteristics that resemble to the plants. The scientists who study the fungi are called as mycologists, and these are in the form of hyphae. And hyphae is a cell in multicellular form or a single form. But these are the pure eukaryotes. These are the topics that we have to discuss today, but I cannot complete the fungi a little bit more. If you have any question regarding the virus and bacteria, you can ask me.
Perfect. Thank you,
Okay.
Professor Elias. That was very clear.
Yeah. Thank you, Elias.
Guys,
thank you.
thank you for joining us today. I shared with you in the comments the upcoming schedule. In the next email that you get from us, you'll receive both the reminder for the next class. It might be when it starts, and under that, we're also going to share with you the tentative schedule. So the other, tutoring sessions, the other Zoom meetings that we have planned, you'll also be able to see them there so you can kind of plan. But the idea is that every day at least we should have some tutoring session at this time at 10 in the morning, Eastern Standard Time, US. So thank you guys. Thank you, Professor Elias, for joining us today and
Thank you.
presenting this material.
Okay, thank you.
Thank you. Thank you, Teacher Elio. Thank you,
everyone.
guys.
Ciao,
Thank you,
ciao.
Teacher Elias.
Have a great day.
Bye. Bye,
Teacher
Jose.
Elias,
Okay.
bye.
Thank
Bye.
you, Mayerlin.
Bye. Goodbye.
Thank you. Bye.